Well, good morning or good afternoon, everyone. So this is Tara. So what we're going to discuss today is the regulations that apply to dietary supplement contract laboratories that are testing for dietary supplements. So the primary regulation for this is 21 CFR 111, which is the FDA Code of Federal Regulations, which is entitled Current Good Manufacturing Practices in Manufacturing, Packaging, Labeling, or Holding Operations for Dietary Supplements. So some people will refer to them as the 111 regulations, and some people will just call them the Dietary Supplement GMPs. So what a dietary supplement is a product that is taken by mouth that contains a dietary ingredient. And in the Dietary Supplement Health and Education Act that passed in 1994, the FDA defined a dietary ingredient as a vitamin, mineral, herb, botanical, amino acid, enzyme, organ tissue, glandular metabolite, extract, or a concentrate thereof. There are multiple dosage forms that can be used for a dietary supplement, including the typical tablet and capsule, also soft gels, a gel cap, liquids, liquid capsules, and powders. But bars can also be considered a dietary supplement, provided that they do not represent a conventional food or a sole item that's used as a meal replacement for the diet. Good manufacturing practices are a system of procedures and documentation, written or analytical, to ensure the product produced has the quality it is supposed to have. The little c, or the big C, depending on how you were trained, for the GMP implies that all the systems that are utilized to comply with these GMPs have to be current. So that's what that C stands for. So the GMPs for dietary supplements, 21 CFR 111, was actually passed in 2007. So it's been a decade since then. So technology changes pretty quickly in a decade. So things that might have been top of the line then to prevent contamination, mix-ups, and errors may not be adequate by today's standards. So the FDA is not going to reissue these regulations. We have to make sure that we're always staying current on technology. So the quality of the dietary supplement is defined by 5 things: identity, purity, strength, composition, and limits of potential contaminants. They also have to ensure that they meet GMPs. If they are not compliant with GMPs in regards to their manufacture, packaging, labeling, and holding, they are all considered adulterated underneath the Act. 21 CFR 11170 talks about specifications, and that's those 5 categories that I just mentioned to make sure we're meeting those. And then in the next section of the regulation, 21 CFR 11175, we have to test products in accordance to those specifications that are listed in 11170. So this is where the quality control laboratory comes in to, one, create and establish those specifications and then test in accordance to those specifications. 21 CFR 11170 and 21 CFR 11175 are the most cited observations that the FDA issues for both warning letters and for 43 observations. In fact, over the last 6 years, since 2010, when the regulations were applicable to all companies, over 55% of the warning letter citations and 43 observations were related to these 2 sections of the regulation. So the quality control laboratory is vital to dietary supplement operations. So this laboratory will provide analytical or microbiological testing, as well as physical testing of the dietary supplement itself and the ingredients therein. It will also do in-process samples and even some environmental samples. Other services that the laboratory would provide would be doing method development for analytical test methods, as well as validations of microbiological methods, and this would incur preservative effectiveness and preparatory testing. Some laboratories will also provide storage of stability samples and then conduct that testing. This lab can be in-house or an external, or most likely a combination of both. So there are sections subparts A through P of 21 CFR 111, and subpart J, which is shown in bold here, is the section that pertains to laboratory operations. Ironically, this is one of the smaller sections of the regulation, even though anyone that has anything to do with the laboratory knows that there's an awful lot involved in the laboratory. That said, All of these here that are shown in red, all of these subparts do reference testing and laboratory operations. And then if we expand that to everything that is applicable to the QC laboratory, that would also include personnel, manufacturing operations, and batch records, because we still have to do testing in regards to those parts of the regulation. So pretty much all of the parts of the regulation apply to dietary supplements. Just note subpart A is the general provisions, which is basically just the definitions. Not only do we have 111 that we have to comply with, but there are also other statutory provisions that we must comply with. So one that is usually most notable, particularly in the laboratory, is 21 CFR 11, which is electronic records and electronic signatures. So anytime we use any sort of electronic system or software, we must ensure that that complies with those parts of the regulation. Dietary supplements are a category of foods. So 21 CFR 101, which is on food labeling, applies to how that label is created for dietary supplements. Some types of dietary supplements can also fall into the categories of low acid canned foods, acidified foods, juices, and seafoods. So these may also come into play. And if we're using a new dietary ingredient, which is defined as an ingredient that was not marketed prior to October 15th, 1994, which is when the regulations were first drafted, then we also have to comply with 21 CFR 190, which talks about those notification processes. And finally, there is the Dietary Supplement and Non-Prescription Drug Consumer Protection Act, which came out in 2006. And this requires serious adverse event reporting for all non-prescription dietary supplement products. So we're going to specifically focus on subpart J, but we're going to reference in some of those other subparts as well and see how these all are commingled. So subpart J is entitled Production and Process Control System Requirements for Laboratory Operation. And this is basically summarized in 5 general sections. So this is 101.303, which talks about written procedures, 111.310, which talks about the laboratory facilities themselves, 111.315 talks about laboratory control processes and the requirements for quality review of those. 320 talks about scientifically valid test methods, and 325 talks about documentation systems. So we're going to tackle each one of those. So first, we're going to talk about written procedures. So although it's not specifically mentioned in 111, one of the most important written procedures that any laboratory can have, regardless of what type of testing that they're doing, is some sort of a chemical hygiene plan. This is a written program that describes the procedures, equipment, and work practices that are there to protect the personnel from the hazards that we have in the laboratory. So this includes hazardous chemicals, this can be biohazards, carcinogens, or other types of toxins. So safety is critical in the laboratory. So there will be requirements for using certain protective equipment or PPE, protective workspaces such as hoods. Hazardous and biohazardous wastes will both be generated, so those have to be identified, used appropriately, and disposed of in an appropriate manner. And then we have to deal with emergency responses. So every laboratory I've ever been in, and I've been in a lab since I was 18, every lab I've ever been in has had some sort of emergency issue. Hopefully these are just generally minor. So we have to have written procedures in accordance to 21 CFR 111, but it never calls them standard operating procedures, but that's what we call them. So SOPs, our standard operating procedures, are critical documents. These serve as the guidance for company policy. They will help standardize the system. They maintain continuity among all employees. They reduce confusion and uncertainty, and they're used for a vehicle for training and instructing employees. So we will have a number of SOPs in the laboratory. So an SOP template should be prepared, and this will have a header that has your company information and the title of the document number, a revision number, because these will go through a change control process. So anytime these were to be modified, we keep track of the previous versions of these documents. There will also be an effective date. And the effective date is not the date that was signed by the last person. It's after the date after which everyone has been trained. And this is the appropriate SOP to be utilizing. Page numbers, And then there'll be categories. So some of the general categories that you'll see are purpose, scope, references. This is referencing other SOPs and referencing other documents. Can also be referencing forms, definitions where appropriate, responsibilities, what groups are responsible for what, the procedure itself, which should be the main portion of the SOP, where things are documented, who authorized this procedure, and the revision history is also very important. It's really nice to be able to go
Tara, sorry to interrupt. I don't know if you can hear me, but we seem to have lost your audio.
Oh, there I am. Is that better? Sorry.
Better.
Should I go back to the previous slide or am I good on this one?
I think just start at the top of this one.
Okay. Sorry, everyone. Technology is great, isn't it? Okay, so one of the types of SOPs that you'll have are on good laboratory practices, and this will actually be a wide number of SOPs. So good laboratory practice is a quality system of management controls for laboratories. This is designed to ensure uniformity, consistency, reliability, reproducibility, quality, and the integrity of the tests that are being conducted there. So this is not to be confused with 21 CFR 58, which applies to clinical laboratories and the requirements for a clinical laboratory and the trials that are being done there. These systems actually came about into the US in 1978 and actually were present in other areas of the world prior to that. There'll be standard operating procedures on the laboratory systems. So everything that's being done in the laboratory will be underneath good manufacturing practices. So there will be a procedure that governs everything that is done in the laboratory. This also includes procedures for equipment and instrumentation, as well as test methods. So for the equipment and instrumentation themselves, there will be SOPs that govern them. And if we jump back to Subpart D of the regulation under 21 CFR 111.27a, all equipment has to be of appropriate design, construction, and workmanship to enable them to be suitable for their intended use. So this is accomplished by doing a qualification, which is often referred to as a DQ, IQ, OQ, PQ, where the DQ is a design qualification for a new piece of equipment. Installation qualification is the installation of the equipment to make sure it's consistent with the manufacturer's requirements. Operational qualification to make sure the instrument operates, and performance to make sure it meets the technical performance aspects that it needs to. So a DQ, IQ, OQ, PQ should be done for all major equipment, and that has to be requalified periodically if there's any major repairs or significant changes to that piece of equipment. And this includes relocating the equipment. So you can't just take an HPLC and decide, oh, I'm going to move it over a foot. If you do that, you may now impact your installation of that piece of equipment or even the operation of that equipment because suddenly you're below an air duct or something. So you have to make sure that those things would be repeated anytime you make some significant change to a piece of equipment. So all of these would be described in an SOP and typically is documented in what's called a qualification protocol that would be executed. 21 CFR 111.27 also in Subpart D then goes on to say that all equipment must be calibrated before its first use and at a frequency that's necessary according to the manufacturer or determined by your use to maintain accuracy and precision. Typically, this is done during the PQ portion of the DQ, IQ, OQ, PQ, but some equipment doesn't require a qualification, and this is usually instruments and equipment that are more mobile, such as a pH meter or even a balance that can be relocated. And the difference is that those are calibrated each time that they're used, or at least daily. So those types of equipment are calibrated directly and don't necessarily require a qualification. So once we've got qualified and calibrated equipment, we have to make sure that we maintain that equipment. And that's talked about in 27C and D to make sure that we're maintaining, cleaning, and sanitizing all that equipment and replacing things as necessary. So there will be a host of procedures describing all this, and it does dictate in the regulation that we have to have logbooks to demonstrate these calibration and maintenance activities. Test methods also have to be dictated into an SOP. So a test method will detail every point, step, and stage of the test method that has to be performed. So this applies to internally developed test methods as well as compendial test methods. Even if it's a compendial test method out of the USP, you still have to put it on your own letterhead into an SOP. This applies to all analytical and microbiological procedures. The format for a test method SOP is similar to what you would see for a standard SOP, but usually there's some additional categories that are added, such as chemical safety, a list of reagents, standard and media preparations. It's really wonderful to have examples of data for spectra and chromatograms or pictures of different bacterial morphology, example calculations, and then any data reporting guidelines. Other types of procedures will also be dictated by SOPs. So these are kind of your subordinate systems, so your protocols, such as the qualification protocol that we already mentioned, method validation, which we'll talk about in a few minutes, and also stability study protocols. Some people will also use a subset category of SOPs, referred to as written instructions. These are typically used when you're using an electronic system and there's a lot of instructions about click on this, point on this, pull down this. And so sometimes those are used for those types of procedures, but they're not necessary if it's covered in the SOP itself. The laboratory will have a lot of other types of controlled systems, such as a sample submission form, often referred to as an SSF, a chain of custody, and standard media or chemical preparation forms and data review checklists. Depending if the laboratory is internal, the laboratory is often the ones that are responsible for raw material and finished product specifications, because that is your technical group. So they are often responsible for that. If you're using a contract laboratory outside of your own facility, then you'd have to provide that to them, and they would not likely have those procedures. So then moving to the next part of 111.310 talks about laboratory facilities. So this could be an entire building for a laboratory, or it can just be a room or multiple little rooms. So any of those have to be suitable in size, construction, and design to facilitate maintenance, cleaning, and sanitizing operations. They have to have adequate space to prevent contamination and mix-ups. Now, these are coming from Subpart C of the regulation on the physical plant, and this seems a little strange to sometimes apply this to the laboratory. Now, what we're doing here is we're trying to protect not the product because it's not the samples that are coming to the laboratory that are being tested are not going to ultimately get to the consumer. However, mix-ups and contaminations in the laboratory those can be critical, because if those are incorrect, if we've got contamination and mix-ups happening in the laboratory, then we're going to release on incorrect data, and we have no data integrity. So these things also apply to the laboratory. So the chemical hygiene program also we talked about before for safe operating conditions. And then the laboratory will have various sources of water. All water that's used in accordance to 21 CFR 111 has to meet the national drinking water standards. Sorry about that. I don't know what's happening. There we go. Sorry. All water has to meet the national drinking water standards, but certain types of methodologies will also require different types of water. So this could include distilled water, deionized water, or even ultra pure water. So depending on the types of methodology that are being employed. The laboratory will have all kinds of different glassware and equipment. So I've just listed a few examples here. So there'll be general equipment, balances, pipettes, centrifuges, heat blocks, that sort of thing. There'll also be safety equipment, fume hoods in the analytical lab, biological safety cabinets in the microbiological lab. There'll be temperature controlled devices, ovens, refrigerators, stability chambers, and there'll be a wide variety of physical equipment depending on what the laboratory specializes in. There'll also be more sophisticated instrumentation, microbiological automated biosystems, for example, total organic carbon analyzers, various spectrochemical types of equipment, such as an FTIR, UV-vis equipment, an atomic absorption, inductively coupled plasma, chromatographic systems, both gas and liquid or thin layer, and various types of detectors to go along with all of that types of instrumentation. Laboratory controlled processes must be reviewed and approved by the quality control personnel in accordance to 111.315. Now, quality control laboratory are quality personnel, but what this is stating is that there has to be a secondary review. So just like we would do for anything else. So one person does the work, a second person has to review that work. And there are a large number of laboratory control processes that take place. We've already touched on some of those. So we have to have appropriate personnel. So in Subpart B, which is talking about personnel, each person engaged in quality control operations must have the education, training, or experience to perform their assigned job functions. So we have to have an assigned job function. So this is typically given in a job description that says this is the background that this person needs to do this job. So it could be a chemist, could be a technician, could be a microbiologist, could be a stability specialist, could be a method developer. So whatever their job is, they have to have a job description associated with that. So people come to a company with a certain amount of education and a certain amount of experience prior. So we have to have either a CV, a resume, or some sort of biography that says, okay, this is what they came to us with, and then we have the on-the-job training that happens at the actual facility, at the actual laboratory. So this will include all of your SOP training, training on equipment and instrumentation, and training on all the various test methods. There'll also be an extensive sample management process, especially if you're using a larger contract laboratory. Some of these larger contract laboratories will receive over 1,000 samples, sometimes thousands of samples every single day. So the receipt and identification of those and a chain of custody for those samples is vital to make sure that there's not some contamination, cross-contamination, or mix-ups with any of those samples. There also should be a specific retention period for those samples and appropriate disposal of those types of samples. There's a lot of chemicals that are present in a laboratory, so we want to have an inventory of all these things. Most states also actually require the fire department will have requirements to know what you have in a laboratory facility. So you'll have some identification of those, history of the receipt, tracking, handling, expiration dates. Expiration dating is really important in the chemical laboratory. Something that goes out of expiration can become very dangerous in the laboratory and can cause explosions and things like that. Our history is riddled with all types of laboratories that have had issues like this over the years. Chemical preparations, everything is tracked back to these chemical preparations, so we have to have some sort of a lot number assignment and tracking of those, so we know that any sample we can track back what preparations were used to analyze that sample, and this all goes back to our data integrity. This also applies to standards, of course, so this would be both chemical and biological samples. There'll be a lot of detail and a management system on those, as well as chemical preparations, and the same applies to so for microbiological media preparations. And it's very important, especially in the micro area, they're using the aseptic technique, and so it's critical that they are not cross-contaminating any samples from themselves or from the laboratory itself. All of the media that they receive has to be demonstrated to be sterile, so not already contaminated, yet it has to be appropriate to promote the growth. So if we're looking for E. coli, for example, we have to have media that would support the growth of E. coli. So if it's there, we're going to see it. So that's called growth promotion testing. In the microlab, there are also other types of controls that happen a little bit later. So during the analysis of samples, there will be routinely incorporated positive controls and negative controls that are run with every single set. There will also be preparative testing that's done to make sure that the test runs for the particular type of sample that we're analyzing. In the analytical world, there are a variety of different types of laboratory controls or quality controls that are used. I've just listed a few of them here. Most of you are probably familiar with the system suitability. that's used for chromatographic analyses. There's also different linearity requirements for that, check standards, the use of blanks, drifts. If you're talking about contamination testing, the detection limit and the quantitation limit are also very important. What the laboratory does is generate data. So there has to be a lot of systems around controlling that data and reviewing that data to make sure that it's accurate and precise. So there'll be a data review process. Typically, you'll see at least a 2, usually a 3 or 4 tiered process. And it depends on kind of how you name that or the nomenclature that you provide. So the analysts will review their own work. There should be a peer review by a person above that or that is trained on the same procedures, and then there's typically a supervisory review. If the laboratory is in-house, there's also usually a quality assurance review that happens with that as well, and that also happens in some contract laboratories. So that would be if you define it in that way, that would be a 4-tiered system, but you want at least at minimum a 2-tiered system, and different people are going to be reviewing the data for different things. Then the managing that data is important. So we need to make sure we're reporting that appropriately, there's been no transcription errors, and we're tracking that data and archiving it as required. So I mentioned very early in the talk that for non-prescriptive, there is a requirement to report serious adverse events for dietary supplements under that Non-Prescription Act that was passed in 2006. That requires that any data that's associated with the potential serious adverse event is maintained for 6 years. So you have to make sure that you're archiving data for at least that period of time. And that is longer than what's dictated in 21 CFR 111. So this is an example why we have to know all of the statutory requirements that we have to comply with. The laboratory will also have a quality incident system. So namely the out of specification investigations. Having OOSs are inevitable in a laboratory, so those should be split into analytical and microbiological OOS investigations. And the reason that those should go down 2 different tracks is because the way that you investigate one of those is going to be different. So you don't expect micro-contamination to be uniformly distributed across the lot or even across your sample. So the way that you're going to methodically evaluate those samples is going to be different than when you would be doing that in an analytical, typically in an analytical setting. There are deviations in the laboratory. We're all people, and so this happens sometimes unintentionally, but sometimes it is intentional. So we need to make sure we have a system to deal with that. And then to correct and prevent any of these types of investigations if we find that there's been some sort of error that's occurred in the laboratory. A really good laboratory will have a very low error rate. So less than 1% is very typical of a very strong laboratory. And then of course we're going to document all of this. If it's not documented, it didn't happen in a GMP world. So we have to have a system to manage all these documents that are being generated. So how they're used, when they're changed. So this is your change control system, your revision control system, and how they're archived. Once we archive a procedure, we don't throw it in the trash. You have to keep it. And remember, we have to keep it for at least 6 years if it's associated with a serious adverse event. Then we have scientifically valid test methods that are dictated to be used in 111.320. Interestingly enough, the FDA did not define scientifically valid test methods in Subpart A, which is our definition section, the general provisions of the regulation. However, in the preamble, which is if you print it out on an 8.5 by 11 pieces of paper, it's over 820 something pages. On page 204, the preamble, which is basically the idea of why the regulations were generated the way that they were, it's the FDA's thinking on all those things. They said that a scientifically valid test method is based on scientific data or published results in scientific journals, textbooks, references, or other types of proprietary research. They go on to say almost 200 pages later that the FDA defined or explained that a scientifically valid test method is 4 things: accurate, precise, specific for its intended purpose, and consistently does what it's intended to do. Accuracy, of course, is the closest of a test result to the true value. This is assessed using fortification samples or spiked samples of a known concentration and being able to recover those back out. Precision is just being able to do that many times. So this, and having agreement among those, and that's usually expressed as a % relative standard deviation. A specific method is one that has no matrix interferences, no instrument interferences, and no potential degradants from, if it's a sample's on stability, that would then later interfere with the analysis of those samples. Ruggedness is the reproducibility obtained with the same sample using different analysts, different equipment on different days, and this is all intended to simulate actual environmental conditions, and that's where the consistently does what's it intended to do comes from. So some of the resources for these scientifically valid test methods can be the USP. They have many methods that can be applied to dietary supplements in their Green Book or the Dietary Supplement Compendium. The AOAC is also an excellent resource for analytical test methods. And if we're talking about flavors and some of the other excipients that we use during the manufacture of dietary supplements, some of these methods are provided in the FCC, or just the Food Chemical Codex, or the Brown Book, if you're familiar with that term. For microbiology, the BAM, or the Bacteriological Analytical Manual, is typically what's used for initiating those methods. And no one knows how to better do pesticides than the EPA. So the EPA has also a number of test methods. Then we must document all of this. So again, if it's not documented, it didn't happen. And if you've not heard this, the GMP term means get more paper, get more people. My personal favorite, because I'm a mountain girl, is giant mountains of paper. All laboratory activities must be permanently documented on paper or electronically at the time of performance. This is mentioned several times in the regulation, so that's critical that it's done as you're conducting the activity. It has to be detailed but concise. We don't want people to write a monologue every time they made a set of standards. If it's handwritten, it has to be legible. It has to be initialed and dated by the individual performing that activity. This is whether it's done manually or if it's done electronically. So this is the audit trail that's necessary for an electronic system that is dictated in 21 CFR 11. Errors have to be obviously crossed out with a single line through, initial and dated, and explained for what the change was. So again, this is where the audit trail comes into play if you have an electronic system. So you can't just cross it out or erase it or something like that. So it has to be obvious that an error was made and it was corrected, and by whom and for what reason. Peer review for accuracy by a second individual or someone of equal or advanced training has to be done. This would also have to be done electronically if we're doing all of this in an electronic system. So this goes back to the training and the data review that we were talking about earlier. So some of the documents that we have in the laboratory that have acceptance criteria will all have to go through a second quality review that we talked about. Some of the different documentation types are SOPs. We talked about those at length earlier. Written instructions, if the laboratory chooses to use that subcategory of documentation system. There will be a lot of laboratory notebooks. Now, these can be electronic or these can be manual notebooks that we actually all had when we were in Gen Chem 101. So these can be handed out individually, so every person in the laboratory would have their own notebooks. There can be one for a particular controlled process. A good example of that is either an FTIR book for doing infrared spectroscopy and analyzing those samples. pH is also another example. Different test methods may have a book that's designed specifically for a type of test method. If you're, for example, running vitamin C consistently, you may have a book that's where you document that you're running vitamin C. Also for equipment, we said that there had to be logbooks for equipment, so you want all those maintenance and calibration activities dictated in that equipment logbook as well. And typically what you see is actually all of these. So there's a combination of all these different types of notebooks being used in a laboratory. There'll also be protocols. We talked about the equipment qualification protocols, so that would document that DQI, QOQ, PQ process. Demonstrating that our test methods are accurate, precise, specific, and rugged would have to be done in a protocol, typically referred to as a test method validation protocol. And when we're doing stability studies, those would also be dictated in a protocol. There'll be a number of controlled forms that are used in a laboratory. I'm a huge fan of controlled forms because they make sure that everyone writes everything down that needs to get written down. It's always in the same place. It makes the review easy, makes us not have to memorize anything because everything's dictated on the form. But forms have to be controlled. Some examples of forms are sample submission forms. If you're using a contract laboratory, they will usually send you those, and now those are all electronic. The chain of custody or a CFC to dictate the life cycle from cradle to grave of a sample in the laboratory. Standard media and chemical preparation, so we have to document when we've done those. So there's usually some sort of a preparation book that's being used or forms that are being used to do that. And then data review. Now again, what the laboratory is doing is generating data. So we want to make sure that that data is accurate and precise. So having a review is important. But a checklist can really make sure that whether I review it or someone else reviews it, we're both going to be looking for the same things. So having a checklist can be very beneficial. And we previously talked about having specifications, whether those are held in the laboratory or held outside of the laboratory. Okay, so I'm available now if anybody would like to ask any questions about any of the presentation, and I thank you all for your attention and attending again.
All right, everyone, if you have any questions, feel free to send them to Tara now. We'll wait a couple seconds to see if anything comes in. Tara, I think you did a great job presenting. No one has sent in any questions. So
everybody knows everything they need to know about a laboratory now, right?
Exactly. So Tara, thank you so much for such an informative session today. We will be uploading this presentation to the EAS website within the next couple weeks. And I hope you'll join us for our next webinar, part 2 of this series, which will be on October 19th at 1 o'clock Eastern. So thanks very much, everyone, and we appreciate you joining us.
Thank you.